All right. Hi, everyone. Welcome to your final exam review, part one. We're going to be going over topics 1 through 3. Do we have any questions before we start about the exam or anything? No? Okay. All right. So we're going to go over, yes. Oh, there, I think it's just multiple choice. It's just multiple choice, but yeah, it's just.
Sorry, Paul, like it's in like 60 points will be in multiple choice and 40 will be like from like.
Yeah, but out of multiple choice. Yeah. Yeah. Any more questions about that? You guys all know when your exam is? Where? And where? Where is it actually?
I don't know.
Okay.
Moral two. Say that again? The dungeon and moral two.
It's 131 and 2.
Oh yeah, that's actually really bad. Yeah, you guys should get there early and get the desks on the sides. That's what I always do when I have an exam there. Okay, so first topic is amino acid chemistry. You guys can raise your hand and ask questions throughout the review if any pop into your head. Okay, so proteins are made up of monomers, and these monomers are amino acids, and they're composed of the same sort of structure. So basically, an amino acid is made up of the central carbon, and from the central carbon, you have the amino group, the carboxyl group, and then you have a hydrogen sticking out of the central carbon, and then you have the variable R group, which basically gives the amino acid its identity, and it's the thing that varies between the different amino acids. So everything except for the R group is considered the backbone. Okay, so the way that proteins are formed is through peptide bonds. So amino acids are strung together in order to form proteins, and the bonds that occur in between the amino acids in order to form a full protein are called peptide bonds. And the way that peptide bonds are formed is through a dehydration reaction. So a dehydration reaction is basically when you have smaller subunits that come together to form an even larger molecule, and water is formed as a byproduct, which is why it's called dehydration reaction. So as you can see here, the hydroxyl group on a carboxyl group of one amino acid gets removed along with the hydrogen of amino acid on another. The amino group on an amino acid of another get removed, form water, and then a bond forms between the carboxyl carbon of one amino acid and the nitrogen from the amino group of another. And then they form this peptide bond. And when peptide bonds are broken, this is called a hydrolysis reaction, and water is used up because you need to re-add that hydroxyl group and that hydrogen back in order to form the two separate amino acids. So does anybody remember why this peptide bond is like a little special? Like what properties does it have? Is it just like a regular single bond, or it is a covalent bond? So covalent bonds mean that the carbon and the nitrogen, they share electrons, right? So it's a very strong bond. Is this peptide bond, does it as strong as, say, this single bond over here between the nitrogen and the hydrogen? There's like a difference between these two. What's the difference? Do you remember? It's what? It's strong. Why? Because well, everything here is the backbone except for these two R groups. Yes. Exactly. So this double bond between the carbon and the oxygen, what happens is resonance can occur. Remember, resonance is where like this double bond can, the electrons in this double bond can go and like sort of move over into this peptide bond, and basically it gives the peptide bond double bond characteristics, right? So it's a little bit stronger than a regular single bond. So we say this peptide bond has double bond characteristics because basically resonance can occur between this double bond and the peptide bond. Any questions? Yes, so double bonds are shorter than single bonds. Yes. Where amino acids are strung together. Yeah, so the ribosome is basically what catalyzes. Sorry. The ribosome is basically what catalyzes the peptide bond formation. Did I just say? Yeah, the ribosome. And so do you guys remember what the ribosome is made up of and what the catalytic component of the ribosome is? Yes. Exactly. So the ribosome is made up of proteins and rRNA. And then the rRNA is the thing that catalyzes the formation of the peptide bonds. Good. So does anybody remember what enzymes that use RNA as the catalytic component are called? Wait, what? Oh, it is? I didn't know that. Well, so close. So close. Right, yeah. A ribozyme. Yes, exactly. Okay. All right, any more questions on peptide bonds? All right, we're gonna go through the different levels of protein structure. Ooh. Okay, so we have primary structure. The primary structure is very basic. It's just the sequence of amino acids in a protein. So the primary structure just could be like, glycine, methionine, cysteine, whatever. And what holds primary structure together are those peptide bonds in between the amino acids. So if I were to make a mutation that switches a glycine into an asparagine, am I changing the primary structure? Why? Exactly. So the sequence of amino acids change. So whenever you see a question that's like, oh, you change this amino acid to this amino acid, what levels of structure are changed? And its primary structure is always going to be affected because it's just simply what the sequence of amino acids is. Yep. Any questions? Okay. Oh, what the? Okay. Secondary structure. Secondary structure, the backbone interactions come into play here. So the backbone, again, is everything except for that variable R group. So the central carbon, the amine group, and the carboxyl group. And so the secondary structure is held together by hydrogen bonds. So hydrogen bonds are basically non-covalent bonds that are formed between partially positive hydrogens and partially negative oxygens or nitrogens. And in this case, the bond that forms is between this partially negative oxygen on that residue carboxyl group of an amino acid and this partial positive hydrogen on that residue amine group on an amino acid. So you can see here in this image of the secondary structure, this is an alpha helix. And you can see that this oxygen on this carboxyl group is forming a hydrogen bond with this hydrogen on the amine group. So when you're looking at amino acid structure, can this hydrogen on the backbone, can this form a hydrogen bond in secondary structure? No. Why? Why? What do you need for the hydrogen bonds? You need partial charges, right? The hydrogen needs to be partial positively charged, right? So let's look at this bond. This bond is between a hydrogen and a carbon. What are their electronegativities? Are they similar? Are they different? They're similar, right? So what is electronegativity, first of all? Exactly, right? So how much an atom is going to like pull electrons towards it, right? So the carbon hydrogen bonds, these are non-polar bonds because they have very similar electronegativities. So the electrons are going to be equally dispersed throughout this bond, right? So therefore, there is not going to be a partial positive charge. So a partial positive charge is what occurs when there aren't as many electrons hanging out next to the hydrogen because another atom, like an oxygen, is pulling more electrons towards it, right? So this hydrogen does not have a partial positive charge because the electrons are evenly distributed throughout this bond since they have equal electronegativities. All right, so this hydrogen, the hydrogen that is bound to the central carbon cannot form hydrogen bonds because it does not have a partial positive charge because it is not bound to an electronegative atom. So let's look at this hydrogen, one of these hydrogens bound to the nitrogen. Can this hydrogen form hydrogen bonds?
Yeah.
Why?
Nitrogen is partially negative.
Exactly, right? Because the nitrogen has a higher electronegativity, so it's pulling its electrons towards it. So you're going to have that uneven distribution of electrons with more electrons hanging out where the nitrogen is. All right. So that is definitely important to know for your exam. Which atoms participate in secondary structure. All right. Any questions on that? Okay. So there are two different types of secondary structure that you can see in proteins. You have alpha helices, which are basically, these ribbon structures, and the amino acids stick out of the alpha helix, and then you have beta pleated sheets, and the R groups stick, they alternate up and down between the sheet. And that's basically all you know for that. All right, any questions? Okay. All right, tertiary structure. The R groups finally come into play. They start interacting, and then you get that 3D folded structure of that classic protein fold. And basically the different bonds that are involved are van der Waals, ionic bonds, hydrogen bonds, and disulfide bridges. All right. So van der Waals are bonds that form between non-polar amino acids. Ionic bonds are bonds that form between full charges, so between a positively charged amino acid and a negatively charged amino acid, or like atom on the R group of that amino acid. And then hydrogen bonds are what we talked about, partial positive hydrogen and partial negative oxygen or nitrogen. And then disulfide bridges are the only covalent bond that we see in the tertiary structure. And they are a covalent bond between what amino acids? 2 cysteine amino acids, right? So it's a sulfur bound to a sulfur on 2 cysteine amino acids. So this is the disulfide bond. Where are disulfide bonds usually found in a folded protein? Yeah, towards the middle. So why does that make sense? Exactly. So it stabilizes the folded protein. It's a very strong bond, right? Covalent bonds are stronger than non-covalent bonds. So it makes sense that it's found more in the center of the protein because it gives it more stability in its structure. Any questions on tertiary structure? Nice. Quaternary structure. All right, so quaternary structure, the same types of bonds that we see in tertiary structure are involved. And it's basically when you have 2 polypeptides that come together and interact. And yeah, not every protein has quaternary structure. Yeah. This one's pretty simple. All right, question. What effect would a molecule that disrupts hydrogen bonding have on protein structure? So if I removed or if I destroyed all of the hydrogen bonds in a protein, what structures will be affected and why? Zach?
Definitely like secondary.
Yeah, secondary. Good.
Pretty sure like one, like this would be affected as well.
Yes. Huh? Yeah, exactly. 2 through 4. Good. Yeah, good. All right. I think we went over this. Tertiary structure is just describing a single polypeptide chain and how it folds and the R group interactions between those. And then quaternary structure is when you have multiple polypeptide chains folded into a 3D structure. All right, domains and subunits. Yay. Okay, so a domain is a independently folded portion of a single polypeptide, and it's so you I feel like I'm gonna draw this. Oh no, this is a good picture. Okay, so this is one polypeptide chain. Okay, are there different subunits in this? No. Okay, good. So let's pretend, I think this looks like it could be folded independently. I don't really know. It would be more evident in your exam if you were given a question like this. But let's say that this is an independently folded part of the polypeptide chain. This would be considered a domain, right? It's folding independently from the rest of the structure. And then subunits are when multiple different polypeptides come together and bind. So this protein is made up of, I think, looks like 4 different subunits. All right, so subunits are just individual polypeptides that are like binding together, and then domains are found within polypeptides. Any questions? I'm sure there will be a question on domains and subunits. I don't know, probably. Everybody feel comfortable with that? Okay, R group interactions. Polar charged R groups, so like between a lysine, this positively charged lysine, and this negatively charged aspartic acid, these 2 amino acids will come together and form ionic bonds. And then if you have the interactions between polar amino acids, like this serine and this threonine, what will likely happen are hydrogen bonds between this partially negative oxygen on the serine and this partial positive hydrogen on this threonine, and then nonpolar or hydrophobic bonds or van der Waals will likely happen between nonpolar amino acids, so like between valine and isoleucine. And van der Waals just basically, you don't need to know like exactly the chemistry of it, but it's just basically when nonpolar amino acids, the bonds in nonpolar amino acid R groups, they have equally distributed electrons, right? Because they're made up of all CH bonds. And so when you have 2 amino acids with multiple CH bonds that come together, the electrons like move sort of in a cloud. And so you have these like very transient partial charges that occur, which then keeps them bound together. So that's what van der Waals is. Okay, so this is a chart that I like because it basically describes how not all of the, for example, if we're looking at the nonpolar amino acids, they are not all equally hydrophobic in this in the same way. So for example, this valine is, or this leucine is a lot more nonpolar than this tryptophan, because this tryptophan has this polar hydrogen nitrogen bond in it. All right, so this shows basically that there's like a hydrophobicity scale. So when you're given a question that's like, oh, leucine and tryptophan, which amino acid is more likely found on the outside of the protein and which amino acid is more likely found on the inside of the protein, what you need to be able to do is look at the different bonds in the R group of these two amino acids and basically say which one is likely found on the inside and the outside. And you guys have the notes chart. I would put this in your notes chart, this scale, because basically if you see isoleucine and tryptophan, where is that? Tryptophan is here. This scale is saying that leucine is more hydrophobic than tryptophan. Therefore, leucine is likely found in the middle. So basically this scale you should put on your cheat sheet sort of thing, not to answer the question, but to sort of like double check. That's why I like this chart, just to double check that you actually know which one is more likely found on the inside and the outside, but you have to be able to exactly describe why this is. So let's see, leucine, its R group is made up of fully carbon-hydrogen bonds, which are nonpolar, right? So that's a nonpolar amino acid. And then tryptophan has these really large carbon-hydrogen rings. However, it has this polar hydrogen nitrogen bond that could maybe form hydrogen bonds, and therefore it's more likely to be found on the exterior. So yeah, any questions on this scale? That makes sense. So basically for those questions, which one's more likely found on the interior, this scale is helpful just to check in, but you have to be able to determine based on the chemical characteristics of the amino acid, which one's likely found on the interior. All right, disulfide bonds, we already went through this. Disulfide bonds are covalent bonds that only occur between two cysteine molecules, and they are found in tertiary and quaternary structure. And yeah, they're very strong bonds, usually found in the center. Yeah. So like thermophiles, these are like organisms that need to be able to live in really extreme temperature environments. Their proteins have a lot of disulfide bonds in them. And that's because disulfide bonds are very strong and are more resistant to denaturation from like really extreme temperatures. So that's a cool fact. Bond summary, you can look at that later. This is just basically what we went over. Okay, which of the following amino acids would be bound to an which of the following amino acids would be bound to an amino acid with a positively charged R group in tertiary structure? Yes. Yep, exactly. Glutamic acid, it's negatively charged. What sort of bond will it form with the positively charged R group? What kind of bond? Ionic bond. Good. Alpha helices and or beta pleated sheets are found in every protein. What characteristic of their bonding makes this possible? So why does every protein have secondary structure? Exactly. So secondary structure is just backbone interactions, and all amino acids have backbones. Good. Oh, waste model proteins. Okay, so you have the ball and stick / wireframe model, and this basically just shows every single atom and covalent bond. So you can see where the backbones and the different R groups are. You have the space fill model that shows every atom and the space it fills basically, and you can see the overall shape of the protein and its surface. This is not useful if you want to like see the different R groups or like the different secondary structures involved, but this is good to see like the overall shape and morphology of the protein. Then you have the ribbon model that shows secondary structure. You can see the beta pleated sheets and the alpha helices, but you can't see the R groups. And then you have the backbone model, which just shows only the path of the peptide backbone. And it doesn't show the R groups. Oh, whoops. Okay, so Skye's gonna lead us into protein folding. And no, misfolding.
Okay. All right. So the main ideas of our protein folding and misfolding. So Rosella's kind of talked about this a little bit already, but we have the importance of temperature on whether or not bonds are stable. Other factors involved in this are pH and salt concentrations and light energy, or like your electromagnetic spectrum. And the cell has a specific optimal range for all of these conditions. So it has an optimal temperature range, an optimal pH range, and salt concentration. And these things can all have an impact on your non-covalent bonds. If you go outside of this optimal range, your proteins might misfold, and this could cause abnormal function in the protein, which may have a toxic impact on whatever function it's supposed to have. When a protein unfolds, that's what we call denaturing. And having a protein that's misfolded or unfolding, again, can be really harmful to the cell. So we have different responses in order to try and fix this. So non-covalent bonds are weaker than covalent bonds. For the purposes of this class, that's what we say. These bonds are your van der Waals, hydrophobic interactions, your electrostatic interactions, ionic bonds, and hydrogen bonds. So your covalent bonds, they can also be broken and disrupted, but it requires a lot more energy for this to happen. So your polypeptide backbone, which is made up of covalent bonds, that's a lot more stable than, say, the hydrogen bonds in your secondary structure, and the disulfide bridges that might be in your tertiary and quaternary structure are also a lot more stable than, you know, the hydrogen or ionic bonds that would be in tertiary structure. So if you're asked about environmental factors, just assume that covalent bonds are strong enough to remain intact, unless you're being told specific conditions that are very extreme and you have to talk about, you know, covalent bonds breaking or something like that, but she probably wouldn't go into that. Everyone wake up. Okay. That was a test just to make sure. So the different environmental factors that we have. So thermal energy basically just regulates like how much your bonds are vibrating. So if you have a lot more thermal energy, that's going to cause more bond vibrations. And depending on the type of bond, this could make it more stable or unstable. So for example, when you're cooking eggs, this is a very common example that we like to use in class. Your egg will shrivel up because the proteins are denaturing. And when this happens, you have the hydrophobic core of the proteins being exposed to this aqueous environment, which they don't like. And so the nonpolar amino acids that make up the hydrophobic core, they're going to find each other and aggregate and clump together. So can anyone tell me why those nonpolar amino acids are clumping together? Why do they aggregate? Yeah. Exactly. Yeah. So the whole concept of hydrophobicity is, you know, water fearing, and it's thermodynamically unfavorable for them to be exposed to this aqueous environment. Laura likes to talk about this Goldilocks hypothesis that she made up. This is not a thing in science. It's just a good analogy that she likes to use, because we don't want to ascribe whether something is good or bad to every situation. Different proteins have different situations that are optimal for them. So some proteins want, you know, more rigidity. So they want less thermal energy because their bonds will be more stable. Other proteins, it's better to have more flexibility. And so they might have an optimal temperature range that's a little bit higher. So again, just because temperature is higher or lower doesn't mean that the protein will necessarily function better or worse. It's very context dependent. So you have to really think about it in the context of the question that she's asking. And this is because the cells have evolved mechanisms that work for them to function best. So we function best at around 37 degrees Celsius. The thermophiles that Rossella mentioned function better at way higher temperatures. Anytime. So temperature is the big one that we talk about, but that's not to say that like pH, salt, and wavelength are not important as well. So pH and salt concentrations can also affect your electrostatic and ionic bonds. Cells, again, have a certain pH and a certain salt concentration within them that's optimal for the function of a protein. So if you throw one of our, you know, like proteins that work in our stomach, the enzymes that work in our stomach, they are optimized for a much lower pH because they have to work in in more acidic conditions, right? But then anywhere else in our body, those proteins would not be able to function in our stomach acid because that's way too acidic for them. And then high energy wavelength, this could even be like you have gamma radiation, right? That could disrupt non-covalent bonds, but it could also disrupt covalent bonds as well if it's really intense radiation. So again, if the intensity is so high that it's able to not only disrupt all non-covalent bonds, it could also break covalent bonds as well. But we focus on breaking non-covalent interactions for the most part. So if you have misfolded proteins, what does the cell do to help resolve this? There are different mechanisms that the cell has to try and fix any sort of protein misfolding. In the cytosol or nucleus, proteins are sent to the proteasome, and this is called the heat shock response. And then proteins going into the endomembrane system, which John is going to talk about later, they have a specific stress response called the unfolded protein response. A lot of neurodegenerative diseases are caused by misfolding proteins and the formation of aggregates. So some examples of this are Creutzfeldt-Jakob or mad cow disease. And the reason for this is that when proteins aggregate, they become insoluble. So again, their hydrophobic core is exposed. They have to form aggregates, and that makes these clumps. And this can have toxic effects, like especially in our neurons, in the case of these diseases. So in order to help prevent misfolded proteins from aggregating, we have what are known as chaperone proteins, which are upregulated when there are unfolded proteins present in the cell. These are recruited to help proteins fold, and it helps stabilize them when they're in this unfolded state so that it's more favorable for them to refold and go into their normal conformation. The chaperone proteins are most likely what is sensing the misfolding. And there's just an analogy here that like if you were on a field trip, your chaperones supervise the kids, your proteins are the kids in this case. But yeah, basically you have this unfolded protein, the chaperone comes along, binds it, helps stabilize it, and this like encourages it to enter its stable conformation where it's folded properly, and then it will release the protein and it can hopefully go about their normal function. Does anyone have any questions so far? Question.
Question about beyond
Relates these to 8, 9, and 10.
yeah, like
You're gonna have to remind me what sections 8, 9, and 10
Yeah,
are.
wait, are you talking about like the different topics?
Yeah.
Topics, yeah.
Okay. It will have a little bit of everything.
Divided into the different topics. So the one tomorrow is going to be topics 4, 5, 6, right?
Yeah,
And then the one on Saturday is going to be 7, 8, 9.
I'm not sure, but this review session is only going over topics 1 through 3. I don't know if that was made clear to you guys. Yes, they're all going to be recorded and posted online. But yeah, we break it up because even though there will be an emphasis on the later topics, there will definitely be stuff from earlier topics as well, especially things like protein misfolding, because that is related to like everything throughout. A lot of these concepts carry over. Yeah. Yes. Yeah. Or I think 8, 9, and 10. Yeah. I'm not sure. All the information is on the bottom of Moodle, and we can remind you guys of that at the end. Yeah. And yeah, everything is on Moodle, and we'll tell the TAs to like send out reminders for when their sessions are. But yeah. As far as
Sorry if that was not my
yeah. Sorry guys.
but
Yeah, so because especially things like protein unfolding, those really carry over into other topics, because Laura likes to ask about loss of function, gain of function mutations all the time, and having a good foundation for understanding those topics is very important in being able to answer open response questions fully. So
even the multiple choice is mostly application based.
Yeah. Yeah, and the multiple choice section is a great opportunity where Laura can go back and like throw, a question from topic 2 or topic 3 at you, because it's just one multiple choice question. So everything will be present. Okay. So does anyone have any questions about what I've talked about so far, chaperone proteins?
Do they also have like some percent like protein you can basically like
If there is a protein that can't be refolded, then it will likely become ubiquitinated, and then that will be sent to the proteasome. The chaperone protein is not the protein that causes the ubiquitination, though, or ubiquitination. Chaperone proteins, I'll talk about it later on, but there will also be like changes in levels of transcription and translation for different proteins, right? Because if you're upregulating your chaperone translation, that means that, you know, there's a problem in the cell. You want to help resolve that. So you're going to lower everything else that's happening in the cell in order to be able to fix that. But I'll go more into detail on that in a couple of slides. So in the heat shock response, this is, again, your cell is sensing that there are misfolded proteins present, and so you upregulate your chaperone production. So you're going to, you know, have more transcription translation of those chaperone proteins. This is happening in your cytosol and nucleus. And again, this is kind of the question that you were asking about, how it's getting tagged by ubiquitin if it can't be saved and then sent to the proteasome. So yeah. And then the other mechanism that we have for helping unfolded proteins is called the UPR, or the unfolded protein response. This is in the endomembrane system, so it's an ER-specific stress response. We sense unfolded proteins in the ER, and then we have a couple of different effects that happen throughout the cell. So we're decreasing translation because, again, if there's something wrong in the cell, you don't want to keep transcribing and translating proteins because they might just fall into the same trap of misfolding, and then you have an even bigger problem on your hands. So you're decreasing your translation, you're increasing the production of chaperone proteins, so those will be upregulated because you want them to be able to help stabilize any misfolded proteins that may be present. And then you're also increasing lipid synthesis. And the reason for this is that the ER is made up, it's a membranous organelle, right? It's just one big continuous membrane, essentially. And so you want to increase the volume of the ER because you're increasing the amount of chaperone proteins that are present in the ER. So you need to increase ER volume. And then in order to get rid of any misfolded proteins, it's got to be sent out, essentially, and there's no proteasome in the ER, so it has to be retrotranslocated out of the ER so that it can be sent to the proteasome. So it has to be removed to the ER and then sent to the proteasome, which is just an extra step versus if you were just in the cytosol. Yeah. Yes, I believe so, right? Because if you're having a bunch of misfolded proteins in the cell, you don't want to keep translating proteins as well. So yes, it's also in the heat shock response as well. Does anyone have any questions on this? Yeah. Yes. Yeah, that's a good question. It's overall translation. So you can have specific transcription factors that target the transcription of the genes that encode your chaperone proteins, right? So you can have targeted transcription of chaperone mRNA, and then that will be translated. But overall, there won't be as much transcription and translation of the rest of the proteins that you have in your genome. Does that answer your question? Okay.
Making more
because the ER is made up of just, it's a membrane, right? So you have like this big membrane, it has lumen space in between, but it's essentially just one continuous membrane. It's actually like connected to the membrane of the nucleus as well. So if you increase lipid synthesis, those lipids are going to go to the ER, increase its volume, and then you can have more, it will accommodate those proteins. Any other questions? These are all very, very good questions. Okay, so now we're going to talk about protein ligand interactions and regulation of proteins. So allosteric regulation is when you have a molecule that binds somewhere to the protein or to an enzyme other than its active site. This will cause a change in the structure of the protein. And again, one of the biggest emphasis, that's not grammatically correct, but what is really strongly emphasized in this class is that structure equals function. So if you're changing the structure of a protein, you're changing its function. We can modify proteins after they've been translated. So this is post-translational modification. This helps localize these proteins to where they need to be active in the cell. So for example, you have ubiquitination, acetylation, phosphorylation. These will all either like change the localization or activity of the protein. And then you also have nucleotide binding and hydrolysis. These are your enzymes that are like ATPases, GTPases, and the binding of ATP, GTP will change the function of a protein, could activate it, deactivate it, etc. Yeah, and then hydrolysis is just the breakage of the second and third phosphate between them. So that's how you release like an inorganic phosphate from an ATP. When it changes from ATP to ADP, you're releasing a phosphate, and that is a highly energetic reaction, which allows you to drive forward a lot of reactions in the cell. So allosteric is when you, again, have a molecule that's binding to a site other than the active site on the protein. This can be either activating or deactivating. So you can have, like, if you have an allosteric activator bind, this is going to increase the function of the enzyme or the protein and, you know, just make it more active. Or you could have an allosteric inhibitor, which in this case is known as a non-competitive inhibitor because it's not binding to the active site, it's binding elsewhere. And the binding of an inhibitor will, again, change the conformation of the protein so that it's not able to bind to its ligand. Whereas, again, if you have an activator, it's making that binding to the ligand more optimized. So you're going to have increased binding affinity to your ligand in that case. Does anyone have any questions on these? Okay. Yeah, just general conformational change in protein. It can either be activating or inhibitory. So post-translational modifications that we can have, these usually change the surface chemistry of certain proteins, and that will cause conformational changes, could target them to different areas of the cell. And again, we've already said, like phosphorylation, ubiquitination, that can just be added on to the protein. And then you have the nucleotide binding, which again, ATPases, GTPases, they break the bond between the second and third phosphate, and they're coupled to a lot of different reactions in the cell to help drive very endergonic reactions, which are energetically unfavorable. They drive forward those reactions because this is a very energetically favorable reaction because it's very exergonic. Are there any questions on this at all? Okay. So just a quick review of KD and like the math behind this. This is a way to measure your protein ligand interactions, and the KD is your dissociation constant. So in this equation, you have your protein concentration, ligand concentration, and then your complex concentration. The complex is the ligand and protein that are bound together. So it's protein times ligand over the complex. The way that you go about measuring this is that you set a constant amount of protein and then you add increasing amounts of ligand. And then the concentration at which the amount of free protein is equal to the amount of protein that's in complex. So when P equals C, essentially, that's the half-bound state, and this is your KD. I feel like this is sometimes a tricky thing. So does anyone have any questions on that? This might just be a good thing to write down on your note card so that you don't forget what it is. It's not quite as intuitive as everything else that we talk about. All right, now enzymes. So we use enzymes to catalyze all sorts of reactions in the cell. They help speed up these reactions. and they are regulated. So we already went over this, but ribozymes are a particular type of enzyme that are made up of protein and RNA. And remember that the RNA is what catalyzes the reaction in this particular type of enzyme. Rossella went over that already. Okay, so the way that enzymes work, and I think it's always really useful if you can understand a reaction coordinate diagram. I think it's very helpful to you because it has everything, like all these words are just in this image right here, right? So you have your activation energy, which is the energy between the reactant and the transition state. The transition state is a very unstable molecule. It's not something that you can like see in real time at all. And because it's very unstable, that's why it's at a higher energy level than either the reactant or the product. And the way enzymes function is that they help lower this activation energy because it makes it more favorable to reach that transition state. It's at a lower energy, and so it's easier to reach there. It's like, you know, you're trying to climb up a mountain, and it's way easier to climb up a little hill than it is to climb Mount Everest, right? So enzymes only affect activation energy. They do not change the energy of the reactants or the products, right? The overall delta G of the reaction is not changing. Only the energy of your activation energy is changing, the energy of the transition state. So that's just something to remember. And can anyone tell me if this graph over here is endergonic or exergonic? Why?
Because you actually stored it, yeah.
Yes, exactly. So if this were an endergonic reaction, you would just see that the products are at a higher energy than the reactants. This would have a positive delta G, which is energetically unfavorable. And so that's an example of why you might need to couple to ATP hydrolysis, because that's very exergonic. It releases a lot of energy. And when you couple something like that to an endergonic process, it helps move that process forward. Any questions on reaction coordinate diagrams, how enzymes work? Okay. So we talked about the dissociation constant, which is your KD, and that's all related to enzyme affinity. And this will change how well your enzyme essentially binds to substrate or product. And you want this was one of your problem set questions a while back, but again, you don't want to have a high affinity between your product and your enzyme, because once your enzyme catalyzes a reaction, you want it to release that product. If the product has high affinity for the enzyme, that enzyme is now kind of stuck, bound to the product, and it's not going to continue catalyzing the reactions. It might in fact catalyze the reverse reaction instead. So under normal circumstances, the enzyme has a higher affinity for the substrate than it does for the product. The product can sometimes be an example of an allosteric regulator that we talked about. So it could be a non-competitive inhibitor of the enzyme. And so once there's a high concentration of product, it might go and bind to the enzyme and shut it off. And does anyone know what type of regulation that's called? When you have the product of a pathway binding to an enzyme and shutting it off? Yeah. Yeah, exactly. It's negative feedback. When you have, you know, again, an end product that's going and binding to the enzyme and causing it to decrease the amount of, decrease the flux through the pathway, that is your negative feedback. And now John is gonna talk to you about protein trafficking. Are there any questions about anything that I said? I know it's a lot. We have a lot of practice questions for you guys at the end. That's why we're running through this kind of quickly. But yeah.
All right. Thanks. All right. So third topic, final topic for tonight, protein trafficking, gene expression by yours truly. Okay, protein trafficking. This kind of starts relating more to the stuff we've been looking at over the past couple of weeks. So it's not like, you know, revisiting old picture books from your basement. So proteins must be trafficked to specific sites inside or outside the cell in order to function properly. Great example of this is insulin. If you take a more advanced biochemistry course, you will be learning about insulin's function in the bloodstream and how it brings glucose into the cells and senses for glucose levels. But it's a good example of a protein that needs to be translocated out. And another example is DNA polymerase. And as we've been talking about DNA stuff these past couple weeks, it must be in the nucleus in order to function. And eukaryotic cells use signal sequences in order to localize proteins to specific sites. And we will start looking at that right now. Okay, so there are two different types of ribosome locations. You can have free roaming ribosomes in a cytosol, and then you can have ER-bound ribosomes. And the difference is where the proteins will end up. So free roaming ribosomes, the translation will occur and the protein will be already in the cytosol, so it is easier to be translocated elsewhere. Whereas the ER-bound ribosomes will translate proteins straight into the ER lumen. And so they are bound for the EMS or endomembrane system. And that is the major difference. So what parts of the cell are included in the EMS? So the ER, Golgi body, lysosome, cell membrane, and the transport vesicles that move between them. So if any protein has to be in the EMS or has to go through the EMS, it must have an ER localization sequence. And if it needs to stay in there, it needs a retention sequence, and we will talk a little bit about that. So that's a picture of the EMS. Signal sequences. So common misconception is that these sequences are post-translationally added to these proteins. They are part of the primary structure actually, so they are not post-translationally added. They're already in there beforehand. And so it tells proteins where its destination is going to be. So NLS, ER, there's different ones. And so the ER localization sequence and the nuclear localization sequences are the ones we talked about the most. And so if it has an ER localization sequence, it goes to the ER. If it has a nuclear, it goes to the nucleus. And if it has a mitochondrial, it is sent to the mitochondria. Pretty straightforward. So close up for the ER localization sequence. Once it has that sequence embedded in its primary structure, it is bound to the EMS and will stay in there until the protein is secreted. And if it needs to stop anywhere, it will have a retention sequence for that specific location. And the ER retention sequence, like I just mentioned, if it needs to stop anywhere, it will have such retention sequence. And so if it needs to stay in the Golgi body retention sequence, cell membrane, cellular membrane, retention sequence. So these are the different signal sequences. I think it's not space filling enough for you to like put in your note card. So I would try condensing it a little bit more, but it's good information to have. So there's a couple differences that are key. For the nuclear proteins, they already are folded in a specific way in order to enter through the import cycle that we will talk about in just a little bit. Whereas for ER proteins, they enter unfolded. And for the mitochondrial proteins, they also enter unfolded. And keep in mind, the last row is where the signal sequences are located in an amino acid fashion. So if it's N-terminal, C-terminal, so the ER is N-terminal, and the mitochondrial is also N-terminal. And then the nuclear is on the outer surface of a protein. So it just means it is very easily recognizable. It has, like, if a protein needs to be in the nucleus, it has to be in the nucleus. And so the nuclear, oh yeah, there's an exception to that, to the primary structure thing that I told you guys. For the nuclear sequences, they are added on. But that is why they are not, they are found on the outside of the protein. They have to be added on because it is like essential that you recognize where that protein in the nucleus needs to be. But that's the only case. So these will also be sent out. I think this stripe specifically is like really good, but it could be condensed. All right, so question. Protein is made to replace a receptor on the cell membrane. This protein was translated by a ribosome bound to the ER. So if you imagine that this protein underwent a mutation where the retention sequence is deleted, and if we are assuming that the protein's ER localization sequence is still intact, where will this protein end up? Why? Very good. Good job, Brian. I didn't bring any. Sorry. Okay. Nuclear import. I've gotten a lot of questions about this, and I don't know if these charts are going to be on the exam. A tip I would give you guys is to, regardless of it being on the exam or not, like the chart specifically, get familiar with the drawing, be able to like explain to each other what each step is. They're like pretty easily broken down into like 5 different steps for import and 7 for export. So as long as you're able to like talk about each one to like a friend, your cat, fish, someone on the phone that you're trying to about, whoever, be familiar with this. And I also say this for the signal transduction pathways. I think that is very important because you can look at it during the exam, like if you draw it on your chart, but we tend to lose a couple IQ points during exams because of like stakes, and just be familiar with it. Okay, so the import cycle. First step, important is gonna bind its cargo protein. That's already gonna be folded, and it's gonna have a nuclear localization sequence. So it's gonna bind to important, and then it's gonna go through a pore on the nuclear outer membrane. And so once it gets into the nucleus, there is a process up there on the RAN GDP. And so RAN GDP will turn into RAN GTP because it is being facilitated by a GEF. What's a GEF? And what's it do?
No, it takes the,
Well, yeah,
lets it fall out.
yeah. Very good. Look at you. So proud of you. But yeah, and so GDP will bind to RAN, and then that will cause, or RAN will go in, RAN GTP will go in and cause a conformation of change onto important, which will then lead it to release its cargo. And then once the cargo is released, then it'll go do whatever it needs to do. And RAN GTP will diffuse out of the nucleus, where a RAN gap is gonna go in there and facilitate the or it will switch RAN, or it'll help RAN switch its conformation state from GTP bound to GDP bound. And then once that happens, important comes loose and the cycle restarts. So yeah, so GTP will be hydrolyzed and then important will unbind it. Okay, any questions about import? No, okay. And then export, here's the chart. And so it's like basically the opposite of import. So RAN GEF starts in the nucleus, and GEF is going to bind RAN GDP and then cause a conformational change. And then GTP diffuses into that site, and RAN becomes bind to GTP. Once RAN is bound to GTP, exportin is going to be able to bind to it. And then once exportin is bound to RAN GTP, there's a conformational change, which then allows it to bind its cargo protein. Once the complex is created, then it is able to diffuse through the nuclear membrane through a pore. And once in the cytosol, again, a gap is going to be there. And so the RAN gap is going to bind to RAN, which then will lead to RAN hydrolyzing the GTP into GDP. The gap doesn't do it. It just allows RAN's intrinsic catalytic ability to hydrolyze said GTP, which will then leave a RAN bound with GDP and inorganic phosphate. And then the cargo will be released in the cytosol, and the exportin will be diffused away from the RAN, and then it could just go back into the nucleus through the same port. All right. Any questions about export? No. Oh, you have a question? No. Okay. So gaps and gaps. Justin helped us out with this. So they're general terms for proteins that act on the RAS family of GTPases. GEF, guanine exchange factor, and it facilitates the exchange of GDP for GTP. GAP, GTPase activating protein. It helps activate the intrinsic catalytic ability of RAN, and RAN is a GTPase, and so GAP binds the GTPase, and then sticks an arginine into the active side of the GTPase, which then increases the rate of hydrolysis. I don't think you need to know that specifically, do you? About adding the arginine in? No, yeah, okay. So question, how would nuclear export be affected if there's a loss of function mutation in RAN gap? Yeah.
X.
What won't be able to let go of exporting?
Oh, RAN.
And then what?
Just be like.
Yeah, and so after that, there's gonna be a pool of cargo bound like complexes, and then the export cycle will like eventually cease because you'll run out of exportin that can do it because they're all gonna be bound to cargo. But yeah. Okay, let's move on. Transcriptional regulation. So transcription factors and chromatin. So transcription factors regulate RNA polymerase. They are binding domain or binding to promoter DNA. And so they are basal transcription factor in eukaryotes, sigma factors in prokaryotes. And so there are different types of transcription factors. We are going to talk about positive and negative transcription factors. They are pretty, it's pretty straightforward. Positive upregulates it, negative suppresses it. And so only in eukaryotes do we see chromatin, and it's how the DNA is stored. And so when the DNA is in chromosome format, when it's condensed, it is heterochromatin, and that doesn't allow for any sort of transcription machinery to go in there, at least not as easily. Once it is a little bit looser, then it's called euchromatin, and that's when you have a little bit easier access to the DNA. And so yeah, that's about it. Okay, and then RNA processing eukaryotes. The two specific kinds that we mentioned in class were the 5' G cap and the poly A tail. So the 5' G cap is a covalently added guanosine on the 5' end. And so this doesn't allow for RNA nucleases to go in and chew messenger RNA, and it's added in the opposite direction of what you normally think any sort of nucleotide addition would happen. And so, yeah, it just prevents, the mRNA from being degraded when it doesn't need to be degraded. And the poly A tail is a long chain of adenine nucleotides, and they are chewed over time because they're non-coding material. So you eventually start running out of it. And that is required for export from the nucleus. And so there are two different drawings up there. But it's pretty straightforward. It's just RNA processing in eukaryotes for this class. And then another kind of processing that is very important that we spent a little bit more time on is splicing. So they're on premature messenger RNA, which is the top picture over there. There are exons and introns. Exons are what they are coding, and so they are translated, or not translated. And introns are non-coding, so they need to be spliced out. And the enzyme that cuts out these introns is the spliceosome. And so a question I had was, why are there introns if they're just going to be spliced out? So the fact that we have different introns allows for splicing, but then you don't necessarily need every single exon also in your mature mRNA. So different cells are able to alternatively splice, and so you can get different isoforms of proteins from the same gene in different cells. So you don't get the same form of protein expression depending on the different kinds of cells that are using this protein or whatever function the protein needs to be doing. So that's a drawing over there with different colors that helps it. I don't know, it's just easier to visualize. But the introns will always just get spliced out. All right. So then there's this little flow chart of the different kinds of gene expression regulation in eukaryotes. So there's transcriptional control, there's RNA processing control, mRNA transport localization control, mRNA degradation control, translation control, protein degradation control, and protein activity control. Do they need to know like specific kinds for every single one?
Like an example.
Yeah.
If Corey gives you an example, you should be able to tell, like, is it RNA processing versus, mRNA localization. I think you've had questions like this in class where, like, you get a list of all these options and you be able to, like, pick the questions. I don't know if that's going to be a question on the exam, but, like, since you've been tested on it before, it's definitely there.
Okay. And here, eukaryotes versus prokaryotes. For this class specifically, we like saying that there's no RNA processing. There is RNA processing, you just don't need to know the specific kinds. Versus on the eukaryotes, there's all these different processes that we talked about. Some similarities in the processing is that they regulate mRNA degradation rates, they regulate mRNA localization, rate translation, protein activity and degradation, and transcription with transcription factors. And so a couple key differences between eukaryotes and prokaryotes, other than the fact that prokaryotes don't have a nuclei. So eukaryotes package DNA into chromatin, and eventually chromosomes. Prokaryotes don't. Also a key factor that's not here. A key factor that's not here is that prokaryotes are able to simultaneously transcribe and translate, whereas eukaryotes are not able to do that. And that is a process that we will be talking about. We're going to review it again on the following review sessions, but that's a key factor. And so prokaryotes don't have a nucleus, and so there's no eukaryotic-like RNA processing. But yeah, and then the different transcription factors, basal for eukaryotes, sigma for prokaryotes. And that's about it. We're gonna have some questions also for you guys that Skye so gladly came up with and arranged. But
Yeah, funny.
but yeah.
Okay.
Should we stop recording? Or do we keep recording?
Yeah, we can keep recording.
Are you guys later?
Yeah, we'll send out everything and the questions as well. Oh my gosh. Sorry. I'm trying to make it when?
I'll post it as soon as I can.
Okay. So I'm gonna just try and cover like half of it because we couldn't get a Kahoot to work. So
Do you need me to sign back into something? Oh,
nope, that's the wrong thing. Oh, you don't have it open.
no, I don't.
Oh, I need you to open it.
Yes, you will send it to me first
I did. I should have.
on email.
Just like over there.
Is it S McMorris?
Yeah, it should just be like one of the first things in your shirt.
Oh, I'm in a different account, that's why. That's funny.
Wait, it's gonna pop up the answers though, so yeah, like cover it up. Hi, have a good one. Okay.
Should I just like take the HDMI off?
No, it's fine.
It's fine.
No, because they have to be able to see the questions. All right.
Okay, well,
Alright.
you can figure it out.
Yeah.
I'll just need to share the screen again. That way, just minimize everything else. Oh my god, yeah, you're a CC cleaner, freaking. Okay. Let's block this tab. It's this one. Sure. Okay. Oh,
No,
no,
that's wrong.
that's this one.
That one. Yeah.
Okay.
Okay.
It's sharing the whole screen now, but.
That's fine. They'll, yeah. Okay, so question 1. Name the strongest bond. Okay. We have a bunch of questions just from each topic for you guys to go through. Yeah, wait, okay, everyone look away for a second. What should I do? If I make it bigger?
Just like go like. They're just not, they're not looking.
Nothing. Wait. I'll just
Wait,
yeah,
oh.
because that's why. I told you not to look.
Nothing.
We got it.
Wait, I got it.
How did you zoom in so much?
Because we're trying to hide it. Wait, just move it up. Just move the whole thing
That's
up.
what I'm trying
No,
to
no,
do.
the whole thing.
Oh.
Full screen.
Okay.
Oh my god, dude.
Okay. Okay. Now name the strongest bond. What do we got?
Alright, somebody just shout it out.
Sorry. Okay. Technical difficulties. Okay. The answer is covalent. So the reason that in like biology we say that covalent is the strongest is because ionic is obviously between two charged molecules, right? And in the cell, you have all of these, it's an aqueous environment, you have all these different partial charges that are going around, and that will weaken an ionic interaction. So I know in chemistry, we say that ionic bonds are the strongest, but in biology, because we're speaking in terms of the cell environment, it's covalent because you have this equal sharing of electrons and they're not, like hydrogen bonding with the water molecules in the cell or something like that. Does that make sense? Everyone feel good for that? Okay. Next question. Disulfide bonds are present in which protein structures? Correct. Yeah. Good job. Next question. Which level of protein structure would urea have the largest effect on? Yes. Yeah, be loud, be proud, guys. Okay, secondary structure. Why? It's talking about hydrogen bond. Exactly, and all your hydrogen bonding. Okay, so true or false, secondary structure is hydrogen bonding between our groups. False, correct. It's hydrogen bonding between backbone. True or false? The backbone of a protein would be broken when placed in a solution with a pH of 2.5. What types of bonds make up your backbone? Mm-hmm. Yeah. So what is significant about them being covalent bonds? Mm-hmm. Yes, but think about, I mean, I don't know the exact pH of our stomach acid, but our stomach acid is pretty acidic, right? And we still have proteins that are able to function in our stomach and certain enzymes, right? So it's not strong enough to be able to break our covalent bonds, but you're good thinking with it. Oh, okay, ignore that. This is a problem. Okay. A protein with one polypeptide can contain multiple domains. True or false? Humor me, guys. Humor me. Okay. True. Thank you. Okay. I'm like so scared to stroll. Okay. A domain on a protein in a frog likely to be similar to a protein with the same functions in humans. Why? Give me reasoning.
Not a frog, we're different.
We're different? Are we that different? I mean,
Call me a frog?
so I don't really know. They were trying to, yeah. So true here, because domains and proteins, if it's a highly conserved protein, it's going to be something that is going to be the same. So for example, like DNA polymerase is, is pretty constant throughout all organisms.
Also,
So
also this can be a structure function question. So it's saying that the protein has the same function. And so if it has the same function, that means the structure is likely going to be the same. So it'll have a similar domain.
yeah, you're not going to get asked that. Obviously. Probably not.
You just pulled that out of your head.
No, I did not write this. Yeah. Spoiler alert. Okay. This. Okay, which of the following would be considered an example of tertiary protein structure? Van der Waals interactions between R groups, hydrogen bonds between backbone, amino and carboxyl groups, covalent disulfide bonds between cysteine residues.
Hey, you always choose one.
It's A and C, you're right. Yes, because it's interactions between R groups. So Van der Waals between R groups is still an R group interaction. Okay. Which protein model would you use to show secondary structure? Yeah. Why?
That's model.
Yeah, exactly. You want to show those alpha helices and beta sheets. Okay. See, this is why I have to have it in front of me too.
Nah, stay still. No.
Don't hate on space. Yeah, I would. Fair.
Politics with ribbon as well.
Out of here. Okay, true or false? In an alpha helix, the R groups of the amino acids are available to make interactions with other parts of the protein and or other molecules. Why? Yeah, exactly. Yeah, you need all of your R groups to be sticking out of an alpha helix. Wait, where am I? Oh, okay. Why are R groups of amino acids available to make interactions with other molecules or other parts of the protein? Yeah. They stick out, so. Okay. All right, if you mutate a protein, I think this was one of your like topic quizzes. If you mutate your protein at the 21st amino acid, have you mutated an amino acid closest to the N or C terminus? Yeah. This is because proteins are pretty big, right? So this would be closer to your N-terminus. You have two proteins with the same domain, which is true. What did you say? Yes, yeah. Again, domains are conserved. True or false, a protein of one polypeptide can contain multiple domains. What did you say? Oh, okay. True, yes. Because a domain is just an independently folded part of the protein. Okay. So this is like a multi-part question. There's a mutation that changes the aspartic acid at the 12th amino acid in the polypeptide sequence to glycine. Does this impact primary structure?
No.
Fight about it. Who's right? Right.
I said I suggested 20 hours.
Why? Someone give me reasoning. Yeah. Yeah, the amino acid order is changed. Yep. Okay, what about secondary and tertiary structure?
Secondary doesn't change.
Mm-hmm. Yeah. Glycine is nonpolar and aspartic acid is polar charged. So, and then quaternary structure, and then I'll reveal the answers. And then quaternary structure, yes, no, maybe. Okay. And the reveal. You were right, secondary structure not affected. So the third one is a kind of an if, because we don't know if there are multiple subunits. We're not told this, but if there are multiple subunits, then it may impact the quaternary structure. Okay. Next. Mitochondrial signal sequence is cleaved off after transport, but ER is not. Okay, we said true for this one. It can, your ER signal sequence, it depends. I mean, if it's a retention sequence, it's gonna stay. Yeah, it's not cleaved off. Okay. The mitochondrial signal sequence, you're never exported out of the mitochondria, so that is cleaved off. Okay. A protein can enter the nucleus while fully folded. True or false? True. True, yeah. Because nuclear pore is big enough for that. You have a misfolded protein with an NLS. Will it be able to go into the nucleus?
Thank you.
Bye, have a good one. Bye. You're right. Okay, yeah, because it may not be able to bind important. If it's misfolded, you may not have the NLS on the surface of the protein, which is what important binds to. Okay, next, a protein with an NLS can move back and forth between the nucleus and cytosol. You guys are good. You guys are good. Okay. A protein that enters the ER won't go back into the cytosol. Why? Someone tell me why. What's your reasoning for true?
They just never leave the ER. I think they're made empty.
They are translated into the ER. You're right. You're the correct answer.
Oh, that's what
Okay. All right. Can you explain? It won't go back into the cytosol. Why is that? Yeah, so it may be sent to the Golgi and then somewhere else in the cell. It may be sent out of the cell, but it's not going to go back into the cytosol. Yep. If a protein needs to be secreted out of the cell, it will contain an ER localization sequence. Oh, sorry, there's more to that. As well as an ER retention signal sequence. That changes it. False, yeah, it won't have retention signal sequence because it's going to be secreted out of the cell. Okay, so this is multiple choice. Has an ER localization sequence and a mitochondrial localization sequence. Where will the protein go first? Do you know why? So where's the location of these signal sequences? Yeah, for the ER, it's an N-terminal signal sequence. So it's read first, and so it gets localized to the ER first. Few.
Like a lot.
Yeah, we don't have to go through all of them. How are you guys feeling energy-wise?
Not the highest.
Not the highest? Yeah, we're gonna send this out. But this is kind of up to you.
Question.
Yeah, who wants to keep going? All right, we can keep, we can have a, if this were a democracy, we'd be out of here. Okay, let's answer this question, and then I have a batch of true or false that I would like for you guys to answer, okay?
Okay.
Okay. Yeah. And then you can go. It's not even 9 o'clock, guys. We're doing great. All right. If a protein has an ER retention sequence, an NLS, and a Golgi localization sequence, where will it end up? This is a tricky one.
NLS nucleus, right?
Good job, guys. Yeah, because it doesn't have an ER localization sequence, so it won't be sent to the ER at all. And then it won't go through the endomembrane system. Well, the example that we just had of the mitochondrial localization and ER localization. So, yeah. Yeah. Yeah, this one, I think she's asked this on problem sets and stuff, so it could be on there for sure. It's a pretty common question. Yeah, the ER localization sequence is at the N terminus, so it gets read first. Yeah. I think it's not. I think it's because the ER one gets translated into the ER4. No, because the ribosome is still being taken to the ER, right? But you have, when it's going into the mitochondria, it's in like a half-folded state already. Whereas as soon as that ER localization sequence pops out, it immediately gets translocated to the ER to finish translation. Okay, true or false? The protein enters the mitochondria fully folded. Yeah, it wouldn't fit if it's fully folded, but it's in some sort of, I'm gonna double check on that one for you. But yeah, it's in like a half-fold state or something. Okay. True or false? Proteins start translating when ribosomes are docked to the ER membrane. Yeah. Huh? Yeah, all proteins. This one is false because if it has the ER localization sequence, that needs to get translated first before it's recognized as an ER localization sequence to get taken to the ER. Does that make sense?
Okay.
So, the protein has to, so you have your mRNA, right? And the ribosome starts translating that. The only reason that a protein will be translated into the ER is if there is that ER localization sequence that comes out of the ribosome and then is attached by the transport protein that brings it to the ER to finish translating inside the ER. But translation still starts in the cytosol before it's moved to the ER. Because you need to translate that localization sequence before the cell knows where to take it. Is that clear? Okay. Every time a protein is localized to the endomembrane system, it must have an ER signal sequence. True. Okay. A protein cannot can leave the cytosol even if it doesn't have an ER signal sequence. False, yeah. An NLS is a stretch of positively charged amino acids. True. Okay, we're almost there, you guys. GAPs exchange GDP for GTP in the cytoplasm. Why? Yes. Good job. Yeah, because okay, this is wrong here. And I will fix this. GAPs do not hydrolyze GTP to GDP. Why is this wrong? Not John. Yes. Yeah. They make it faster. What is actually hydrolyzing GTP to GDP? Exactly. Okay. True or false? GEFs exchange GDP for GTP. This is the opposite of what we just said. True. Thank you. Oh. In the site, okay. No, she's never gonna ask that. Even I was just like, yeah, true. It's, yeah. If you're thinking about the nuclear import-export sequence, the GEF is in the nucleus, so that's why. That was a tricky one. Okay, that's all for tonight. We'll post, we have a bunch more questions, but we'll post all of them for you guys. Get some sleep. Thank you, thank you.
Thanks, guys, for doing all that.
Okay.
That was crazy. What?